Pathogenic fungi like septoria are enemies of the farmer, capable of attacking crops and leading to dramatic reductions in yield and quality. The farmer does have weapons at his disposal. Fungicides are effective when used correctly. However, there can be a problem. Fungi are living organisms, and they can adapt to the chemicals we rely on to control them. And because fungal spores are produced in their millions, there are consequently many opportunities for genetic changes to occur. Systemic fungicides work by entering the fungal cell, interacting with the target fungal protein. The fungicide locks onto the protein, preventing its normal function, stopping the fungus in the leaf from growing and controlling the disease. Different fungicides interact with different proteins in the cell. For example, azole fungicides inhibit membrane synthesis in the fungal cell, while strobilurins interfere with energy production. But the fungus can mutate, producing an altered protein so that the fungicide no longer locks onto the target. This stops the fungicide from working, so that the mutated fungus will survive and reproduce. This process is called target site resistance and is the predominant type of change affecting fungicide performance. If the mutation is substantial, it will have an immediate effect on the fungus, turning it from sensitive to resistant. If enough of the mutated cells survive while the originals die, then eventually only the resistant strain is left. Multiply this by the millions of spores that are produced by the fungus life cycle, and it is possible to reach a critical mass of fungal cells that can no longer be killed. When this happens, the initially sensitive population is replaced by the resistant type, and the disease can no longer be controlled by the fungicide. But sometimes the process takes place gradually. A slight change in the target protein may begin to reduce the effectiveness of the chemical. Over time, further small changes in the protein can occur, so that it becomes more and more resistant. When the same chemical is used again and again in isolation, this can select for more resistant types, so that population of the fungus becomes less and less sensitive and eventually can no longer be completely controlled by the fungicide. Increasing the use of the same chemical in an effort to maintain performance can actually increase selection of the gradually mutating fungus spores, leading to a reduction in disease control and loss of yield, and the farmer won't know it's happening until it's too late. Some mutations succeed in surviving the fungicide, but no longer function properly in other ways. These fail the fitness test and die. But with so many opportunities for mutation, eventually some will survive. These may not have complete resistance to the fungicide to begin with, but as their population increases, they will mutate further, and some will have improved fitness. But for the farmer, whose main concern is the crop, this is not a game of chance. Mutations occur naturally all the time, and sooner or later, changes in fungicide target proteins are likely to occur. Careless use of effective fungicide will kill off the sensitive strains of the fungus and actually hasten the day when the resistant strains win the battle. So how to avoid this happening? There are 4 strategies we can use. First, prevention. By using the fungicide before infection, we can prevent the infection happening rather than try to cure an outbreak that's already occurred. Prevention can avoid the really huge number of spores developing in the first place. Second, limitation. Limiting the number of applications will help prevent resistant strains rapidly increasing within a fungus population. The third is mixture. Using a combination of types of chemical helps to avoid a resistant mutation multiplying. If one fungicide doesn't get them, another type will. The final strategy is alternation. Alternating fungicide types provides more chances to kill a mutant strain. If they survive one product, then a different type may well be effective. The stakes are high. With 9 billion mouths to feed by the middle of the century, it is absolutely essential to increase the yield of staple foods like wheat. The loss of an effective fungicide would be a major setback, and only careful use of these vital products can help avoid this happening. Remember, these fungicides are a key weapon in controlling disease and thereby securing crop yields. Developing new compounds is a long and arduous process, and the farmer could find himself left defenseless for many years. This video was produced for BASF in collaboration with the National Institute of Agricultural Botany, NIAB, and Rothamsted Research.